All right. Ooh. Crackle, crackle. All right. So today we're going to keep talking about acids and bases. Okay. So what we've done so far, okay, is we know that we are thinking about how our bodies regulate pH. So we're going to be thinking about both our lungs and our kidneys in this process. And right now we've got kind of a decision tree that we've set up, right? So we're starting with our pH, right? And we know that we can define acidosis and alkalosis by looking at the pH and how it corresponds to our normal range. So if our pH is too low, and too low for us is going to be less than 7.35, that's acidosis. Okay, so that's our definition of acidosis. And on the other side, we have alkalosis if we have our pH above 7.45. So we also know that these conditions can be caused by either respiratory problems or renal problems or things you ate or things you're doing. Okay. So to tell those things apart, there are some other numbers we can look at. So we mostly talked about the respiratory side last time, although we also moved our way into the renal system a little bit. But we're going to start by thinking about respiratory disturbances, right? So to tell if the problem is your breathing, you're going to look at the partial pressure of carbon dioxide. So if you're in a state of acidosis, what you're going to notice if you've got a respiratory problem going on is a partial pressure of CO2 that's too high, right? CO2 is a biologic acid, so too much CO2 is a problem that's going to lower our pH. Okay, so too high, we're comparing it to our normal systemic partial pressure of CO2, which is 40. So this is an indication that you are specifically in respiratory acidosis. Now, we're not going to look at numbers on the other side, but if you have a partial pressure of CO2 that is 40 or even maybe below, right? So a normal partial pressure of carbon dioxide in your blood, okay? Then that means your pH disturbance is coming from something else. So that's going to be metabolic. This is how we're kind of assessing a potential patient and trying to figure out what's going wrong, what we might need to fix. Okay, so on the alkalosis side, we can do the same thing. So our easiest thing is going to be to start by checking that carbon dioxide level, that partial pressure of carbon dioxide. Can measure our blood gases. And if our problem is the CO2, then we're done. All right, so CO2 is an acid. So if we don't have enough acid, that could cause alkalosis, and that would be due to our respiration. So if our partial pressure of carbon dioxide is not lower than 40, then that's not what's causing this pH that's too high. So that's going to be some type of metabolic alkalosis. We're going to have a more complicated version of this flowchart later, but this is the part that we've already done so far. So I'm going to make it easier when we get to that chart in a little bit. Okay. All right. So these are some of the things that we talked about last time. Okay. So we know also, this is where we left off, that we have 3 lines of defense when we have an acid-base disturbance. So when our pH of the blood is not what it should be. So our first is that we can buffer out hydrogen ions. The H in pH are those hydrogen ions. So we can limit their effect by soaking them up with a buffer or squeezing our buffer like a sponge and putting a little bit more of them into the blood. But that doesn't actually reverse our pH change. To reverse it, we're going to need to compensate using either our respiratory system, so altering our breathing, or using our kidneys, which is going to be our renal compensation. So both of those can actually change the acids and bases or buffers in your blood and will actually change the pH itself. Okay, and the pH is under homeostatic regulation. So this is going to be happening without you thinking about it, which is good because assuming that you, like me, have never thought and tried aggressively to consciously change the pH of your blood. Okay. So walking through these are happening at different speeds. So buffering is super fast because the buffers are already just kind of in your blood. So we mentioned hemoglobin, and we've mentioned bicarbonate, which is really our most important buffer, especially when it comes to thinking about the renal system. So ECF there, reminder, is extracellular fluid, and your plasma in your blood is one of our types of extracellular fluid. Okay. So we can buffer out too many hydrogen ions. So you see there are H+, those are our hydrogen ions. Here's our bicarb, our bicarbonate, that's our bicarbonate ion. Right? And here we see how those bicarbonate ions soak up hydrogen ions into our carbonic acid. Now, there are other buffers, right? So we briefly mentioned that hemoglobin kind of acts as a buffer because of its reactions with carbon dioxide. In your intracellular fluid, so inside the cell, you'll have various proteins that can also kind of soak up hydrogen ions acting as a buffer, as well as phosphates doing the same thing. So we have a couple other buffers that can work in conjunction with our bicarbonate. Okay. Now our second line of defense, still pretty quick, but it's going to take a couple minutes for it to really have an effect on your blood pH. But this is actually going to be changing the contents of your blood, or rather than just kind of soaking up hydrogen ions and, making them not react with things even though they're still there, which is what's going on with the buffers. Okay. So respiratory compensation is just referring to the fact that since a lot of the pH of your blood is due to those carbon dioxide levels, you can alter your breathing to alter the carbon dioxide levels, which alters the pH. So more formally, we can regulate the pH by varying our ventilation. So if you increase ventilation, so if you breathe faster, right? If you breathe faster, we think of breathing as pushing out CO2, you're breathing out CO2. So if you have excess CO2 building up in your blood, right, you can breathe faster, breathe it out, means you'll pull your pH back to where it should be, right? Now the opposite also works. Right. So if you decrease your ventilation, if you breathe slower, you're not going to be breathing out carbon dioxide as much. If you're not breathing out that carbon dioxide, it's not going to go anywhere. It's just going to stay in your blood. So you're going to see build up in the level of carbon dioxide as a result. So that's how the lungs can help us. Okay. So we're going to imagine that we've got a situation where we're not going to think about the cause just yet, right? But we're going to think about a situation where our plasma pH is too low. So that's acidosis. So low pH is too much acid, right? We already know that we have peripheral chemoreceptors that can detect carbon dioxide levels. We talked about that shortly before break when we were talking about regulation of ventilation. So these peripheral chemoreceptors, they're sensing chemicals, particularly carbon dioxide is what we're interested in, right? And a low pH, right, means too much acid, means too much CO2. It's a possibility for the cause of that, right? So if you have too much acid, you're going to try to remove acid from your bloodstream. And one way you can do that is by breathing out excess CO2, pulling that pH back up because you've breathed out that biologic acid. This brings the partial pressure of carbon dioxide in your blood back down, which is what's pulling the pH of the plasma back up. Okay. So we want to associate lots of CO2 with a low pH, right? So paying attention to these arrows here. Okay, so this is a negative feedback loop. If we pull our plasma pH back up to where it should be, we're done. We don't have to do this anymore. So our ventilation would stabilize. Okay. So I've got a question for you here. So that's what's happening with the CO2. And as we move into our next little bit, let's think about what other things could change our pH. So if we see a drop in pH in the bloodstream, what else could we do to bring that pH back up to our desired range? So we're thinking about this from the perspective of the bloodstream, right? So from the perspective of the bloodstream, right, if we have a low pH, what we mean is we have too much acid. That's our acidosis. Okay. So the reason these two in green are our two best options, right, is if we have too much acid, we can add buffer to the bloodstream. So add bicarbonate, add more bicarbonate is really what we're going to be thinking about here. And that extra bicarbonate that we're going to add can soak up acid, can soak up excess hydrogen ions. So that would bring the pH back up because those hydrogen ions are not kind of stuck in that buffer. The other thing that we could do is just remove acids from the blood, right? We can take our acids and just grab them, pull them out of the bloodstream. And so that's what I mean by get rid of hydrogen ions. So hydrogen ions are acid, really. Like technically, that's what the pH is measuring. Right? So we could actually pull those hydrogen ions out. And when we're pulling them out, really what we're going to be thinking about, since we're about to be talking about kidneys again, is we're going to think about peeing them out. We're thinking about urination. Okay. Well, that's the job of your kidneys, right? So renal compensation is our third line of defense, AKA the slowest acting here. And this is going to take you hours, maybe days, to adjust your pH using your renal system, using your kidneys. Okay? Now, what the kidneys are doing with respect to acids and bases and pH, they're going to be regulating excretion of hydrogen ions. So they're going to be regulating how many hydrogen ions are showing up in your urine and getting expelled from your body, right? So if you pee out hydrogen ions, they're not in your bloodstream anymore, right? So fewer hydrogen ions in your bloodstream means a higher pH. Okay. And the other thing that we can be regulating excretion of is bicarbonate, right? So bicarbonate is really our main buffer that we're thinking about. If we have bicarbonate in our bloodstream, it can soak up excess hydrogen ions, which would lead to a slightly higher pH. You pee them out, then we're not going to be soaking up all those hydrogen ions, so our pH would go down. Now, your kidneys can also, under certain conditions, make brand new bicarbonate ions, so not ones you ate, not ones you got from metabolic processes elsewhere. You can make new stuff, and we'll talk about how that happens today. The overall effects that we see from increased acidity, right? So if we see an increase in acidity in the bloodstream, we're trying to regulate that, right? To bring pH back up from a state of acidosis, right? Acidosis is low pH. We want to bring ourselves up, right? We can secrete hydrogen ions into our nephrons and ultimately excrete them from the body. So that's going to actually remove them from circulation, bringing pH back up. We can reabsorb bicarbonate. So if we reabsorb bicarbonate, now it's in the blood, it's going to act as a buffer. Right? So we're going to loop back to that first line of defense and buffer out excess hydrogen ions. And synthesis of new bicarbonate would work the same way. If we make new bicarbonate, we're also going to put that into the bloodstream. So this is more buffer to soak up acidity. Right. So a reminder before we talk through the next stuff, we're going to be talking about what's happening in different parts of the nephron. So we're going to be thinking, we have a glomerulus, we have Bloemann's capsule at the front. That's not going to be a topic right now in our acid-base stuff, okay? But that's what this is. Next, we have a tubule. It's kind of twisty. Sometimes we mention that in its name, sometimes we don't. This is our proximal tubule or our proximal convoluted tubule. So remember, proximal, when you saw it in anatomy lab, meant stuff that was like nearer in towards your core. This is nearer in towards like the head of your nephron. The next structure you see is the loop of Henle. This dives down deeper into the kidney. We're also not going to be talking about that today, but we do want to know what nephrons look like as a whole, since we'll be talking about them a lot over the next couple of days. So let's label it. And then we have a distal tubule, also twisty. So sometimes distal convoluted tubule. We're not going to distinguish just distal tubule from distal convoluted tubule. We're just going to write distal. So remember, distal means farther out when you're talking about it in anatomy lab. So here we're farther out, further along our nephron. And then we move into collecting ducts. So collecting ducts collect, filtrate on its way to become urine from many different nephrons pulling together. That's what we got here. So collecting duct. So when it comes to regulation of blood pH, acids and bases, bicarbonate, all that jazz, we're going to be thinking about the proximal tubule behaving in one way. And then we often lump together the distal tubule and the collecting duct, and we will be doing so today. So distal tubule and collecting duct is also a location in the nephron where we're going to see this regulation happening. Okay. So that's our reminder of what those are. So this makes a little more sense. So when we're thinking about how your kidneys handle hydrogen and bicarbonate ions, we can think about what's happening in that proximal tubule. So like the first kind of stretch in the nephron. And what's going to happen there, we're going to find that our proximal tubule overall kind of acts as a bulk reabsorber. Okay. So a lot of things that we see, not just bicarbonate, but also bicarbonate, are going to get filtered into the nephron. So they're going to come out of your glomerulus, which is a specialized capillary. They're going to move into this first part of the nephron. They're going to be in the filtrate. We refer to that fluid that we've filtered out as the filtrate. Okay. But we don't want to pee out everything that we pulled out of the blood. So we're going to reabsorb many things, and including bicarbonate, which is relevant to us today. So when we say reabsorb, we mean we're going to take it from the filtrate and put it back into the bloodstream, back into the peritubular capillaries. So there's basically just there's blood flow running alongside the nephron. Okay. So bicarbonate here is going to be reabsorbed back into the bloodstream. So about 80 to 90% of the bicarbonate that you filter out of your blood, the glomerulus, is going to get reabsorbed pretty much immediately in that proximal tubule, go back to your bloodstream. There are other things happening here in the proximal tubule that relate to pH as well. So we do have some hydrogen ion secretion going on here. So it's not all reabsorption in the proximal tubule. So secretion, we're going to be moving hydrogen ions into the filtrate. So they're going towards the urine, towards eventual excretion. We also have sodium being reabsorbed in the proximal tubule. That's going to be important primarily because sodium gradients drive so much of what's happening in cells. So sodium reabsorption, movement of sodium is going to be helping us transport things as we look at what's going on in the proximal tubule. In the distal tubule and the collecting duct, we're going to be thinking about the same kind of direction, but slightly different ways of doing this. So we're going to see secretion of hydrogen ions again. So some hydrogen ions are going to be moving into the filtrate towards becoming urine and being excreted. That's going to be coupled here towards a different way of getting bicarbonate into the blood, which is that we're going to make bicarbonate in these cells. Then we're going to take that bicarbonate that we make in the cells, and we're going to move them into our bloodstream. So we're going to synthesize or make new bicarbonate ions, putting them into the plasma here. We'll take a look at what that looks like. Okay, so this first picture is talking about specifically the proximal tubule and some epithelial cells. Okay, so we got a location and we got a type of cell here. All right. So there's a lot going on. We'll do it piece by piece. I'll highlight. Yeah, okay. We'll walk through it step by step. Okay. So what's going on here is we have our tube and our tubular fluid and our filtrate here in yellow. So that's our tubular fluid. This is our peritubular fluid. So peri just really means next to. So fluid next to the tube, and your bloodstream would be here. Okay, so this is where our blood is. So that's going to be important for orientation. So for directions, if we're thinking about reabsorption, reabsorption is going to be movement towards the blood here. Okay, so moving back towards the blood is reabsorption. So what we're trying to understand in this diagram is how we are reabsorbing bicarbonate. So reabsorbing that buffer so that we keep it in the blood so it can keep buffering out hydrogen ions. Okay, so here's our bicarbonate that we're looking for. You can see it's moving towards the blood, right? So here we have that end point of our bicarbonate reabsorption here. Okay. So let's track where that came from. Right. So bicarbonate is one of the things that's getting filtered out with a ton of stuff. Most of the contents of your plasma is going to get filtered out at the glomerulus at the very beginning of the nephron and the renal corpuscle, so the glomerulus and Bowman's capsule. Okay. So we already have bicarbonate swimming along through the tubular fluid, right? So we can see it here, this filtered bicarbonate. Okay. So in order to reabsorb this, we're going to take that filtered bicarbonate and we're going to convert it into water and CO2. Okay. So the way we turn it into water and CO2 is we're going to combine it with a hydrogen ion that we have in the tubular fluid to make carbonic acid. The Ca here is our enzyme carbonic anhydrase. In this case, it's embedded on that apical membrane. So the apical membrane is the one pointing towards the tubular fluid. The basal membrane or the basolateral membrane is the one on the side facing the blood. Okay, so we're converting bicarbonate and hydrogen ions to carbonic acid, then converting them to water and CO2. Okay. So when we then allow CO2 to diffuse into the cell, which it can do because it's a nonpolar gas, it doesn't need a transporter here, we're essentially taking those bicarbonate ions in. So here we see that CO2 and water have moved easily into the cell. Okay. Now we're basically just going to do this in the reverse, right? So we're pulling in bicarbonate ions by turning them into carbonic acid and then water and CO2. Now we're going to take water and CO2 that we have in our proximal tubular epithelial cell. We're again going to use carbonic anhydrase to do the opposite reaction. So here's our carbonic acid, which we can then peel apart, dissociating into hydrogen ions and bicarbonate ions, right? So it took us a couple steps, but we really just got those hydrogen ions and bicarbonate ions that we had in the tubular fluid into the cell. Okay. Now, we do think of CO2 as an acid, but we should notice here that those hydrogen ions that we end up with in the cell, they're getting secreted back into the tubular fluid, right? So those hydrogen ions aren't moving towards the blood. They're moving back into the tubular fluid. So they are going to be in that filtrate as it becomes urine and excreted from the body, right? So we're not upping acid levels by doing this. That's why it says hydrogen ions secreted here, right? That's kind of their source. They're kind of swirling around in a cycle here. Okay. Now those bicarbonate ions do need to be transported across the basal membrane in order to get back into the blood. So we have to use energy is really what I'm saying here. Our source of energy is a sodium gradient, right? So our sodium gradient here is caused by our sodium-potassium pump, right? So our sodium-potassium pump creates a situation where we have low sodium inside the cell, and we have higher sodium levels in both the tubular fluid and in the peritubular fluid. And you can tell that in part because of the colors of these arrows in this figure. Red is always against the gradient when your textbook makes these figures. All right, so we can see sodium moving high to low on that apical membrane. And here we see our sodium-potassium pump. That sodium is moving from low to high. Okay. Now, as we move across the basal membrane, we're going to be doing secondary active transport, moving sodium and bicarbonate over here. Okay. Okay. So in the distal tubule and collecting duct, we have a little bit of an adjustment to make. So we have a similar thing, right? We are still setting up a system where we are moving bicarbonate ions towards the blood, right? Okay. But the source of those bicarbonate ions is different, right? So what you'll notice here, now that we're in the distal tubule and collecting duct in a type of cell called an intercalated cell, right? We're not pulling bicarbonate out of the tubular fluid in this diagram. What we're doing is taking carbon dioxide that we have in that cell, right? So carbon dioxide that's in that cell, either because it diffused in from somewhere or because it was made through metabolic processes, right? But not because we took bicarbonate and converted it in the tubular fluid. Otherwise, this process is working pretty much the same, or at least the same for our purposes, right? So we're converting it to carbonic acid, dissociating into hydrogen ions and bicarbonate ions, pulling those bicarbonate ions towards our bloodstream, and taking those hydrogen ions out into the tubular fluid where they will be secreted. Another thing that you might notice on this diagram is that those hydrogen ions over here are combining with another molecule in our tubular fluid. So what we're doing here, you don't want the pH of your urine to be too low. Like that would be painful if you had really acidic urine. So we are going to also buffer out some of those hydrogen ions in the tubular fluid in the urine, right? So that we don't go below a minimum pH in our urine. Minimum pH of the urine, you don't need to know, but I think it's about like 4.5. So you can have acidic urine, but not super, super acidic urine. The final thing that we may do in renal compensation as a defense against major changes in pH is use a molecule called glutamine as another source of bicarbonate ions, essentially. All right, so here you can see that we take glutamine, and glutamine, when it breaks down, one of the things it breaks down into is bicarbonate ions. Here we see the other thing it's breaking down into is like ammonia. Okay. So here we're producing brand new bicarbonate ions from the glutamine. And again, we're just going to pull them into the blood. All right. So all of these mechanisms that we're looking at with these directions are telling us how we would use our kidneys to compensate for probably respiratory acidosis, right? So we're trying to fix acidosis in all of these diagrams. Okay. So we're pulling more bicarbonate into our bloodstream here from a brand new source, the glutamine. We can see that we're taking hydrogen ions and we're secreting them out, combining them with that ammonia. Okay. So glutamine is an important kind of extra step, an extra way that we can compensate for major pH changes, particularly major acidosis. So we're going to be regulating hydrogen ion secretion through this, right? So our hydrogen ions, right? We know that bicarbonate reabsorption is happening most of the time under normal conditions, usually those first two figures that we ran through. So the normal reabsorption of bicarbonate in the proximal tubule combined with that synthesis of bicarbonate in the distal tubule and the collecting duct is usually enough to keep us in our regulated range as far as pH goes. But in severe acidosis, we're breaking glutamine down, producing brand new bicarbonate, extra buffer, and peeing out extra hydrogen ions as we're secreting those hydrogen ions with the ammonium. Okay. Okay. So summary to this point, right? So for our pH, when we're thinking about what balance of carbon dioxide and bicarbonate ions keeps us in our range, we want a pH of around 7.4. We got a little flex of about 0.05 on either side of that. This is going to correspond to a ratio of 20:1, so 20 bicarbonate to 1 carbon dioxide. Okay. In acidosis, we have a ratio that is too low. So it can be too low because we have too much carbon dioxide or because we have too little bicarbonate, too low buffer. Alkalosis is the opposite. So if our ratio is too high, that indicates we don't have enough carbon dioxide or we have too much buffer potentially, right? So we can compensate for these disturbances using either kidneys or lungs, depending on what our problem is. If we're doing it with the kidneys, we're regulating the bicarbonate side of this ratio. If we're doing it with the lungs, we're regulating the carbon dioxide side. So when we're thinking about compensation, right? So we have respiratory acidosis. Respiratory acidosis, really the cause of that is going to be hypoventilation. So you're breathing too slow, too shallowly. You're not breathing out the CO2 that's in your blood. So we know that your systemic arteries should have a partial pressure of CO2 of 40. Your veins, as they're returning to the heart and going out to the lungs before they get to the alveoli, is at a partial pressure of 46, right? We should set that partial pressure back to 40 as we return from the lungs. If you're breathing too slowly, too shallowly, that's not happening. So you're going to be carrying over some of that carbon dioxide, and it's going to end up in your arteries, leading to that higher partial pressure of carbon dioxide in the systemic arteries. So when we see those increases in partial pressure carbon dioxide levels due to chemical reactions that are going on in your blood, balances of carbon dioxide and hydrogen ions, this essentially is going to get converted into higher quantities of hydrogen ions and lower pH. So that's our problem, our respiratory acidosis. If the problem is that you're not breathing right, good chance you're not going to be able to fix it by fixing your breathing because we've got something going on with your respiratory system, right? It shouldn't be doing that. It shouldn't be causing respiratory acidosis. If you could control it, hopefully your peripheral chemoreceptors would be sensing that and already altering your breathing rate. Okay. So if something's going on with your respiratory system that's causing acidosis, you're going to try to compensate for that, so make up for it using your kidneys. So renal compensation is when we're trying to adjust the pH of our blood because of a situation caused by the lungs, creating respiratory acidosis. So if our pH is too low, that means we have too many hydrogen ions, too much CO2. So our two options are to pee extra hydrogen ions out. So we will secrete hydrogen ions into tubular fluid and then excrete them out of the body. And we're also going to bring new buffer into the bloodstream to try and combat that excess of hydrogen ions in the plasma. So we're going to reabsorb more bicarbonate. And we also now know that we can also synthesize new bicarbonate, pulling it into the plasma as well. Now on the opposite side, our other problem, respiratory alkalosis, can be caused by hyperventilation. So hyperventilation, you're breathing too fast, you're breathing too much, maybe you're having a panic attack. So you're breathing out a lot of carbon dioxide. We're actually seeing a drop in our CO2 levels. So as we have a drop in CO2 levels, we have a corresponding drop in our hydrogen ions. This means our pH has gone up. So alkalosis, pH went up due to low carbon dioxide, low hydrogen ions. So to adjust for that, to compensate for that, to do renal compensation to adjust for respiratory alkalosis. We're going to try to not secrete so many hydrogen ions. We don't want to pee them out. We want to keep them in the bloodstream. So we're going to slow down or decrease our hydrogen ion secretion. And we don't need as much buffer. We don't want to be putting more and more of that sponge into our plasma. So we're also going to slow down how much bicarbonate we're reabsorbing into the bloodstream and how much bicarbonate we may be synthesizing and pulling into the bloodstream as well. Right, so that can compensate or adjust that situation of respiratory alkalosis, bringing our pH back to where we want it to be. Now, metabolic acidosis and metabolic alkalosis may be caused by the kidneys, might be caused by something you're doing, right? So metabolic, we're thinking just like anything that's not your lungs, anything that's not respiration. Okay. So we're seeing an increase in the acidity of your blood. That's not because of carbon dioxide, right? If we're in a situation of metabolic acidosis, okay? So because we know it's not a carbon dioxide problem, we know that we should be able to use our respiratory system to fix this. So we should be able to alter our breathing. And if it's something you ate or if it's heavy exercise, then you can use your kidneys to fix this too. If it's because you have kidney disease, that's not going to help you. Okay. So for the respiratory compensation, if you're in metabolic acidosis, right? So metabolic acidosis, problem with hydrogen ion concentrations, not with CO2, but CO2 is still an acid. So if you adjust that CO2 level, you are going to be adjusting the pH of the blood anyway. So what we would do in this situation, we have a situation of acidosis, too much acid in the blood. We want to get rid of acids generally. So we're going to breathe faster, breathe out extra CO2. So sometimes breathing faster is good. Renal compensation, gonna work the same way we've just been describing. Right? So if this is not a kidney problem, then to fix the metabolic acidosis, you're going to increase how many hydrogen ions are getting secreted and urinated out of your body. We're going to increase how much buffer we're putting into the plasma. So we're reabsorbing our bicarbonate. And this is usually when we start to see that synthesis of new bicarbonate happening as well. So this would be like the glutamine part in particular would be kicking in here potentially. Metabolic alkalosis, same thing. Maybe you ate too much baking soda. That could be a cause of metabolic alkalosis, or maybe you have kidney disease, right? So just some reason for alkalosis that isn't your lungs, isn't your CO2, right? So our problem is we don't have enough acid in the blood because we do want some to keep us at that sort of neutral pH. Okay. So we can compensate for that first using our lungs, using our breathing, right? So respiratory compensation, if your pH is too high, what we're going to do is try and match that, right? We're going to bring in extra acid, right? We want to bring in extra CO2 into your blood to adjust that pH because CO2 is an acid and alkalosis means we don't have enough acid. So this is where your breathing is going to slow down. You're going to see decreases in ventilation. They're going to be building up CO2 levels to compensate for this metabolic alkalosis. Now, if the problem is not your kidneys, we are going to try to fix this with our kidneys as well in the kind of longer term, and especially if our respiratory compensation for whatever reason isn't enough. Right? You do have to breathe some. You can't just stop breathing to fix metabolic alkalosis. So in this case, our renal compensation would look like eliminating less acid, right? So you don't want to secrete so much hydrogen, you don't want to eliminate that from your body. You don't need as much buffer in your blood, so you're not going to reabsorb as much. You're not going to make as much new bicarbonate. So that brings us to this chart, which was what I was leading us up to at the beginning, right? So here's how we can diagnose our acid-base disturbances and what kind of compensation is happening. If we are measuring, let's say something in like a blood jar, really, if we're looking at blood gases, right? So to do this, stepwise, we first want to think about, are we dealing with acidosis or alkalosis? So our first question, right, is what is the pH? Right. So 1 is our pH. Right. So this we went through. Too low a pH, you're in acidosis. Too high a pH, you're in alkalosis. So our second question that we're going to try to figure out is what is the cause? Right. So here we can see for acidosis, our problem may be too much CO2, which means we're in respiratory acidosis. Okay. Or our problem might be something else. So another thing that you could actually measure would be our bicarbonate ions, right? So another cause of acidosis could be that you don't have enough bicarbonate acting as a buffer. Our comparison number for bicarbonate is 24 millimoles here. Won't ask you to remember that number. Specifically, I'll tell you if the bicarbonate is too low or too high. Okay. But that would be a metabolic cause. So in this case, our CO2 would not be higher than 40. Now we pull in the compensation piece, right? So for respiratory acidosis, we're going to fix that with our kidneys, renal compensation. So we're going to see that to compensate with our kidneys for this high level of CO2, we're going to need to pull our bicarbonate levels up. So if you're in respiratory acidosis, you're going to see a combination of high CO2 and rising higher bicarbonate levels trying to make up for it. If you're in metabolic acidosis, first thing you're going to do is try and compensate for that with your lungs. So you'll be in respiratory compensation. Okay. So we're going to see CO2 levels changing here, but we are going to see them changing in the opposite direction than if we were trying to diagnose the acidosis. Right? So if you see a too low pH indicating acidosis, but you in fact have really low levels of carbon dioxide, right? Low levels of carbon dioxide should be creating a higher pH. If you're not seeing that higher pH, that means that you're doing respiratory compensation, and that's making up for something else. The other side of this flowchart works the same thing. So I recommend easiest thing is really looking at those carbon dioxide levels, right? So for alkalosis, the carbon dioxide levels are too low, then that's our cause. Respiratory alkalosis, we're going to compensate with the kidneys, renal compensation. If you're in alkalosis, so you're looking at that pH, but you see that your partial pressure of carbon dioxide is really high, that means that the cause was something else, cause was metabolic, and you're doing respiratory compensation. I'll leave you with a final question to answer before you go. So a question about this might look like this. This is how you might be assessing things. So that everybody can leave, I'll just tell you the answer real quick. Okay, so in this situation, pH is too high, so that's alkalosis. Partial pressure of carbon dioxide was lower than it should be, telling us this is a respiratory problem. So we're compensating with the kidneys, our renal system, which is why we see these lower levels of bicarbonate.